Hello everybody, my name is David Yeates. I'm the Director of the Australian National Insect Collection and Curator of Diptera here. The collection is administered by the Commonwealth Scientific and Industrial Research Organisation, CSIRO, Australia's Federal Science Agency. This presentation will give you some information on the Diptera component of the collection, including the staff, some field work we've been doing recently, and some research we've been working on as well. We'll also discuss the new collection building we've been planning and designing over the last few years. The new building will be constructed a few hundred metres from the current site of the collection in Canberra, for those of you who have been here already. My co-authors today are postdocs Keith Bayliss and Brian Lassard. So the Australian National Insect Collection is by far the largest collection of Australian insects in the world. We have estimated about 12 million specimens, most of them, the PIN collection anyways, housed in 3 large collection halls. We have just over 20,000 primary types, that's holotypes and similar. Most specimens are pinned in 23,000 metal drawers. Some of them you can see behind in the background of that slide. We have a staff of about 26 with a similar number of students, retired researchers, and we have 24 volunteers. Actually, in addition to that list, we have a number of staff now contracted to help us move the collection to this new building, which you'll hear more about later in the presentation. We think we have about 1.5 million or so Diptera in the collection, but we haven't counted them, to be honest. Here's one of the ANIC collection halls. You can see it's pretty busy, pretty full. The ANIC is a working collection. It doesn't have a public display function like a museum, and it's been in existence for about 100 years or so. Here's some of the staff in the collection working on Diptera. We have Brian Lassard, a postdoc. His social media handle is Bri the Fly Guy. He's working on stratiomyid and culicid systematics, and I'll show you some of his recent research later on in the presentation. Keith Bayliss is a Schlinger postdoc. He's working on Ecliptrate phylogenomics, and we'll touch on some of his work later on as well. And we also have Juan Lobaton as a postdoc. I'm working with Sasha Mikheyev and Juan on fly microbiomes, and Sasha is based at the Australian National University, which is a few hundred metres from where we are in Canberra. We also have Jaime Flores, who's the Diptera technical support. We have James Lumbers, a PhD student on tachinid systematics, and also Mark Rouleau, a PhD student working on the nutritional value of edible insects in Australia, especially those that are endemic to this continent. Some of the field work we've been conducting recently, they're the red dots on a map of the broad biomes of Australia. You can see that we've worked in rainforests, in open sclerophyll forests, in deserts. A lot of the work we do is with malaise traps, and some of the work, especially that Keith Bayliss does, is at night using mercury vapor light traps, where a lot of interesting things come into the sheets. And that image there in the bottom right is taken during some light trapping on field work at Iron Range in Cape York in far north Queensland. Closer to home, we're also doing some more intensive malaise trapping from Black Mountain Reserve, adjacent to the collection site at the moment, and also month by month samples from more than 10 localities around the Australian Capital Territory, where Canberra is, and New South Wales, the adjacent state. So there's a lot of samples to go through, and some of those samples are providing us with very interesting material for various ongoing studies. Here's some of the recent research we have been publishing and doing, getting ready for publication. This is the results of one of Zwan Lee's PhD thesis chapters, published with a number of other co-authors, many of them who are interested in the systematics of bombyliids. This research was published in Cladistics earlier this year. Zwan combined ANIC and hybrid data with some transcriptome data in order to generate this phylogeny, which is a new view of the relationships of the subfamilies of bombyliids. And he also found that there was a great increase in the diversification rate of bombyliids late in the Cretaceous, which was an interesting finding. And this will give us some great way forward in terms of classifying bombyliids and also understanding where more attention is needed in terms of their relationships. This is another piece of research, this time from Keith Bayliss and a number of colleagues, looking at the relationships of the schizophrene flies, particularly the Ecliptrates. This is work that combines transcriptome, genome, and ANIC hybrid enrichment data sets. And Keith was able to discover a couple of new clades within the Ecliptrates, the cleft pedicel clade, the modified overscapped clade you can see there on that image, on that tree. And I think this is a really new and compelling piece of evidence on the relationships of the Ecliptrates. He discovered that the schiamyzoids are the sister to the rest of the Schizophrena, and that Drosophila and its relatives, the Ephydroidea, are closest relatives to the Ecliptrate flies. And Keith continues to work on additional components of the Ecliptrates using phylogenomic data, and his postdoc on that research was published in BMC Biology in earlier this year. Some more research from Keith there on the left-hand side of the screen there. He's also using phylogenomic techniques, again combining transcriptome, genome, and ANIC hybrid data in order to look at the asteroid radiation, so tachinids and calliphorids mostly, finding a number of well-supported clades in that group, and confirming a lot of what other recent authors have discovered about the relationships of the subfamilies of those families there, but also some new results as well. We hope that'll be published in the next 6 months or so. On the right side of that screen there is a screenshot of I guess it's a tweet from Brian Lassard. Brian's always been very interested in and very accomplished with media presentations, outreach, and social media. And here he's tweeting about the 150 new species that the national collections described in the last 12 months. And of course, many of those were insects. And this is one of the species of Stradiomyides that Brian described in this last 12 months that has a striking resemblance to one of the outfits that RuPaul has worn recently. So that caused quite a bit of interest on Twitter and drew attention to the undiscovered wealth of Australian biodiversity that we have still to work on. Another piece of research there on the right, phylogenomics, again from Brian Lassard. This is a phylogenomic study of the Stradiomyides, the soldier flies. He's working closely with Martin Hauser and Norm Woodley on this, and Keith Bayliss is helping with some of the analyses. But again, this will give us a really good new view of the relationships of Stradiomyides using these very large phylogenomic data sets. And you can see from the branch links there that there's good support for many of the sets of relationships and almost all of the subfamilies there as currently understood on monophyletic. On the right-hand side of that screen, also some more work from Brian Lassard and some of his colleagues who've been working on mosquitoes more recently. This is a paper published again this year in the journal Parasites and Vectors, really announcing the fact that this new vector of Japanese encephalitis, Culex triteniorhynchus, has been discovered in Australia. It occurs up around Darwin in the Northern Territory of Australia, and it was confused with a native species until Brian and his colleagues had a good look at the molecular data associated with those samples. So that's an important biosecurity, has important biosecurity implications and human health implications, of course, once we have the vector of that Japanese encephalitis disease in Australia. Just to go back to the collection itself, we think we have about 1.5 million specimens. It's an estimate. We have about 10,000 species, again, an estimate. Many of them are not described. We have about 3,500 primary types of Diptera in the collection and 70,000 microscope slides and a lot of material, litres and litres of material in ethanol. Some of it has been sorted, but a lot of it is in malaise traps in various stages of being sorted, malaise trap residues. The ANIC has significant collections from Sir J. Paramonov, Dick Norris, Don Collis, Alan Dice, and Peter Cranston over its, I guess, 100 years that the collection's been growing. Some of you may be aware that we've worked on a Lucid Keys to Australian fly families over the past decade and a half. And these were developed and originally released in 2005 on a CD back in those days. It seems quite really old technology these days. More recently, those same Lucid Keys to Australian fly families were released on a website, What Bug Is That? And they've just been released now in a new platform on iOS, for phones and tablets, apps for iOS and Android operating systems. And that has the keys plus information on collecting, preserving and curating flies, fly classifications, and an overview of the fauna, etc. So here's a view of Annex, the current building we're in, Annex Old Building, we're calling it now. It was designed in the 70s, constructed and opened in the early 80s. And it's no longer a world-class standard, I would say. It's too small for the collection now. We're struggling to grow the collection coherently. The building can't be sealed, so it's difficult for us to keep domestids or other pests out of the collection. We have to use a lot of naphthalene in the collection, and we'd certainly like to move away from chemical protectants if we can, so that our staff aren't subject to those while working in the collection. So over the last few years, we've been designing a new building for ANIC. And we've been, I guess, designing over the last 2 years, and we're about to go to tender for the construction, which will begin early in 2022. The architects assure us that the building could be finished in late 2023, and we'll be moving in 2024. We'll be going chemical free. There'll be more and better space for the collection and curation. There'll be specialised spaces for digitisation, brand new molecular facilities, and a trace DNA lab, or an ancient DNA lab. I'm going to be sharing the building with the Australian National Wildlife Collection, a large collection of birds, mammals, and lizards largely. So the collection will be closed for decant and move between 2022 and 2024. At the moment, we're auditing, databasing, digitising, and tissue sampling from primary types in the collection. Here's just a bit of a look at some of the images of the new building from the architects. These are artists' impressions. The outside, brand new meeting rooms and curation spaces, quite close to the current herbarium. And I think my 15 minutes is up. Thank you very much.